Okay, so may I start? So hi everyone. My name is Sofia. I am a first-year PhD student, and today I'm pleased to present our synthesis of our research focusing on elucidating the foraging ecology of these stunning birds that are European Storm-petrels, Hydrobates pelagicus, in the West Mediterranean. So imagine navigating the vast sea, a small bird, less than 30 grams, searching for food. What path would you choose? What influences your choices? Is it the temperature, the swirling currents, or perhaps the wind that guides you? These are some of the questions that we face when trying to understand the foraging behaviors of these birds. So our study aims to first trying to characterize the complex foraging trips these seabirds undertake. Second, we aim to understand which are the oceanographic features that can influence their choices. Third, we need to predict which are the most optimal dining spots for the species, and finally, we need to quantify how well our marine protected areas are covering these spots. So to tackle this question, we initiated a GPS tracking study involving 39 breeding, incubating European Storm-petrels during 3 different years. Our colonies are located in the West Mediterranean, specifically 1 in front of the Benidorm city and 3 in the Balearic Islands, 1 in the northwest part of the Ibiza island, Espartar, 1 in the south of Mallorca, Pobra, and 1 in the south of Menorca, Aire. We employed first-time passage analysis to identify area-restricted search location, or ARS locations, that are indicative of foraging behavior. This allowed us not only to track where the birds traveled, but also to understand when and where the birds were actively searching for food. So, what's the result? First off, cool plots. I know this slide might look a little bit chaotic at first glance, as it includes all the tracking data from all the years of all the colonies layered together. However, these trajectories tell the story of these extensive journeys that these birds undertake. What we're focusing on here is beginning to understand their foraging behavior and beginning to understand where they concentrate the most. We can appreciate their long distance and direct flights punctuated by areas of foraging behavior that are ARS locations. These ARS locations are concentrated across different regions, the most important being the Alboran Sea, one, the North African coast, and the Algerian basin and the Balearic basin. But notice also how some birds make stunning flights, for example, one from Menorca up to the Gulf of Lion, and one from Ibiza, feeding in front of the Ebro River Delta. So now we've just zoomed in on the top 4 foraging regions for Storm-petrels. However, our tracks also revealed some impressive trip characteristics. Indeed, our birds demonstrate remarkable endurance with a maximal travel distance of 1,700 kilometers. Some birds can also venture as far as 400 kilometers away from their colonies. They can also travel at a maximum speed of 80 kilometers per hour, and they can spend nearly 5 days at sea continuously. So now that we've identified where they forage, we want to better understand the environmental influences on petrel foraging behavior. our methodology integrated high-resolution GPS tracking data with near-real-time satellite-derived oceanographic data. This includes the sea surface temperature, chlorophyll A, eddy kinetic energy, bathymetry, and wind. Moreover, as we know that foraging seabirds are attracted by oceanic fronts, we included a level of spatial complexity by calculating the ranges of sea surface temperature, chlorophyll A, and eddy kinetic energy through a moving window function in order to detect differences, gradients in the water masses. We employed a generalized linear mixed model, GLMMs, focusing on the data from Benidorm colony and including the individual as random effect. This model allowed us to explore the complex relationships between several environmental predictors and the foraging behavior that is our locations of the petrels. So what we obtained? First, our findings from the GLMMs showed that the wind, surprisingly, did not have any measurable effect, while the most important variables for foraging storm-petrels were lower sea surface temperature, higher chlorophyll A, greater eddy kinetic energy, and bathymetry. This means that these birds prefer to forage in colder and nutrient-rich and dynamic water with shallow depths. Moreover, the fact that the variables with the moving windows are significant means that these birds are sensitive to local spatial variation in oceanographic conditions. But we didn't stop at Benidorm. The robustness of our model was tested by cross-validating our data with the other 3 colony data. And the model proved highly effective, reinforcing the idea that the identified and selected predictors are consistent across different colonies and across time. Using the validated model, we were able to predict and map the key foraging hotspots across the Mediterranean that you can see as the brighter areas in the map. These predictions not only highlighted the previously identified areas such as the Alboran Sea, the North African coast, and the Gulf of Lion, but also suggested new regions. For example, the Ebro River Delta and the upper part of the Gulf of Lion, such as the Camargue Regional Park, that, if you remember, were the areas where some of our birds concentrated when I showed you previously the animated tracks. It's also interesting to notice that the eastern part of the Gulf of Lion resulted significant, even if our birds not directly exploited that area, but that area is exploited by birds from other colonies, specifically from the colonies in the northern part of Sardinia, monitored by our Italian colleagues, Jacopo Cecere, Federico De Pascalis, and the here present Andrea Benvenuti. And the birds showed to concentrate and forage in that area. The distribution of the most suitable areas for foraging storm-petrels overlap, and this is another interesting part, with regions of higher biological productivity and prey aggregation. So now we try to understand from an oceanographic perspective why these regions are so important. So the Mediterranean is a complex and turbulent system characterized by a rich mosaic of 3D oceanographic structures. The video you are seeing illustrates the mesmerizing dance of eddies across the sea. They are formed by intricate hydrodynamical processes when the Atlantic water enters through the Strait of Gibraltar, giving rise to sub-mesoscale and mesoscale structures that enhance nutrient availability by bringing deeper and nutrient-rich waters to the surface. These processes are also enhanced by the seabed contours, which can focus and intensify upwelling events, further concentrating and transporting nutrient and prey. So the regions highlighted by these eddies coincide with our previously identified foraging hotspots, underscoring the importance of these oceanographic mechanisms in determining key foraging habitats for our species. As we conclude, it's clear that the European storm-petrels depend heavily on specific foraging grounds that are currently underprotected. Despite their ecological significance, key areas such as the Alboran Sea, the North African coast, and part of the Gulf of Lion remain exposed to human threats and are under minimum protection by the MPA's network. Moreover, the current marine protected areas are considered both inadequate and ineffective. The situation calls for a robust expansion of marine protected network, not just in size, but also in the scope, taking into account that the oceanographic mechanism can be both permanent and seasonal. And by focusing on these critical areas, we can ensure a sustainable future, not just for European storm-petrel, but also for our marine ecosystems and the myriad of species that they sustain. Thank you so much.
Thank you, Sofia. Perfect on time. Questions, please?
Hi, Sofia. Thanks for the presentation. I was wondering in the foraging areas if you have found, if you know if there are tuna farms, and if you know, yeah, because I know in other places they associated a lot with the tuna farms, so I was wondering if you are going to do that or if you know.
No, thanks for the question. We did not consider tuna farms, actually. We didn't analyze that tuna farms, but it's something that we consider in next steps. Thanks.
More questions, please? Jose? Ah, okay.
Thank you for a great talk on the best bird in the world. I was wondering how the predictions of foraging hotspots might be changing due to marine heat wave conditions. Any thoughts about that?
Sorry, if they change?
Due to marine heat wave conditions.
Well, of course, they are exposed to marine heat waves, but our predictions are made with a range of years that coincide with the tracking data. So we didn't analyze future marine heat waves, but of course they could be affected by this.
One more, please. Jose. And sorry, is this last one? Okay.
Hello, thank you for a very, very nice talk. I didn't understand how did you end up with, what did you do to predict the foraging hotspots? I missed. After the GLMMs, what was the next step of predicting?
Here, you mean?
Yeah.
Okay, so we made the GLMMs, and we cross-validated our data with the other 3 colonies. And with this, the validated model, we were able to predict of course, we downloaded another time all the variables of all the oceanographic data. Yes, I did not explain it because I did not have time, but if you want, I can explain.
We can discuss after.
We can discuss it later because it's huge work. It seems easy, but...
Thank you, Sofia. And now, Maria Inês with the fatty acid profiles in seabirds, correct?
Yes. So, good morning, everyone. As Miguel said, I'm going to talk about how foraging in different environments can influence the fatty acid profile of two shearwater species, and how fatty acids can tell us more about the quality of marine ecosystems and resources. So here in this map, you can see population density in the Mediterranean catchment area. It is well known that the coastal urbanization is increasing in the Mediterranean countries, and this can lead to habitat degradation, intensive exploitation of resources, and other types of problems like pollution and eutrophication problems along the coast. This graph here shows a lot of pollution hotspots along the coast, and I want you to notice here this small area that is the Alboran Sea that has a lot of pollution hotspots, mainly in the coast of Morocco. This oceanic basin also has other problems like eutrophication and hypoxia issues in their coast, and if we combine all these threats together, we get a cumulative impact model like this one. And here I want you to look again to the Alboran Sea, where you can see there this area is a medium-high impact area with special focus on that orange part there that is close to Nador. It's a coastal city in Morocco that is a pollution hotspot in the Mediterranean and a eutrophication problem area. The Northeast Atlantic Ocean, and more specifically the Portuguese coast, on the other hand, it's a non-problem area in terms of eutrophication and pollution. So, regarding this, we wanted to know if all of these different pressures between the two oceanic basins had some impact in the quality of food resources for consumers, and in particular seabirds. So to do that, we selected two colonetary shearwater species in each basin. Coimbra shearwater, breeding in the Northeast Atlantic and more specifically in Berlenga, that was our study area, near the coast of Portugal, and Spatial shearwater, breeding in Chafarinas Islands, 5 kilometers from Nador, Morocco. So our main goals were to compare the fatty acid profiles of these species and test for different trophic markers which can give information on the quality of resources and potential anthropogenic nutrient intake. Also, we wanted to evaluate the trophic and foraging ecology of these two species and relate them with the bird's fatty acid profile. So, fatty acids play essential functions in the organism, like cell membranes structure, signaling, and energy storage. And part of them can be digested and stored almost intact, which makes them good trophic markers to assess the quality of seabirds' diet. A lot of different studies using different model species have been using fatty acids as trophic markers to assess marine organism diet quality in urban coastal areas with eutrophication and pollution problems. So our methods consisted in capturing birds during chick rearing by night in their nesting burrows and deploying GPS devices. After 1 to 2 weeks later, we collected blood samples and used plasma to conduct the fatty acid profile analysis and stable isotope analysis. So as I said, we followed the birds for 1 to 2 weeks. And after this, after recovering the GPS, we split the data, the foraging trips, according to their dual foraging strategy during chick rearing, meaning they perform short trips to feed their chick and alternate them with long trips to restore body condition. After this, we computed for both species the foraging and home range areas, distribution areas, and statistical analysis show that during short trips, Coimbra shearwaters traveled greater distance over a larger area, and during long trips, they also covered a larger area, but they did it faster and spent less time resting than the Spatial shearwater in Chafarinas. Concerning the fatty acid profile of both colonies, the fatty acid profile revealed higher variability of Aurore shearwaters. This aligns with the wider isotopic niche in the Mediterranean, as you can see. And Scopalis shearwaters, on the other hand, displayed very similar fatty acid profiles, as you can see here, according to the narrower isotopic niche and smaller foraging area during foraging. So now speaking about particular fatty acid groups and markers, we found no differences in saturated and monounsaturated fatty acids. This was expected because seabirds can biosynthesize this type of fatty acids, and therefore we would not expect them to change proportions during the same breeding period and with such close related species. We did not find as well differences in omega-6 and trans fatty acids, which can help sometimes indicate changes in the diet quality and anthropogenic nutrients intake. We found instead significant difference in omega-3 concentrations with higher levels in the Mediterranean. This was not expected since the essential fatty acids like omega-3s are produced by phytoplankton, and in more problematic areas, usually the phytoplankton community tends to change and the levels of omega-3 tends to be lower. However, the Alboran Sea, as you know, is very different from the oligotrophic Eastern Mediterranean Sea, and is also very productive. So maybe for some time the omega-3 levels can be higher than in the Atlantic. Another reason for this maybe is because the omega-3 are very important to stimulate immune responses, and maybe in these areas birds can have a more active immune response to environmental stressors. Other potential explanation is related with diet, maybe a diverse prey availability and selection between breeding colonies can be influencing these values. And we checked for the omega-6 omega-3 ratio, that is an indicator of quality diet, and we did not find any difference between the two colonies, so we think that the quality of the diet was not compromised by these differences. So we also found differences in the odd-chain fatty acids that were higher in Chaffarinas, in Scopalis shearwaters. These fatty acids, along with this other one that I signed here in green, are usually biosynthesized by bacteria, and this can indicate a change in the environment, so in the community of microorganisms in the area. Maybe anthropogenic induced processes and high temperatures near this area might create optimal conditions for the proliferation of these microorganisms. Another significant result was the difference in the concentrations of oleic and vaccinic acids, higher in Chaffarinas as well. These are known trophic markers for urban runoff and waste discharges, and heterotrophic bacteria growing on organic matter. This may reveal more waste discharges in this area, mainly due to the inexistence of water treatment stations in some cities in the coast of Morocco. However, we cannot forget that oleic acid is also very important for energy storage, and high mobilization of this fatty acid might also be related with higher energetic demands of Scopalis shearwaters during their foraging trips, as our results from tracking data suggest they spend less time resting and they take more time to do long trips to find food. For last, and despite we did not find significant difference in the trans fatty acids group, overall, we also detected this fatty acid separating the two colonies. And this type of fatty acids do not naturally occur in the marine environment. Instead, they are associated with industrial food processing and urbanization in terrestrial animals. So in conclusion, our take-home message is that the fatty acid profile of these shearwater species reveal differences between the two environments, with higher variability of profiles in the Atlantic due to a broader foraging range and wider isotopic niche, and differently for scopoly shearwaters. And the presence of certain fatty acids associated with coastal urbanization and pressures highlights a potential environmental impact in the diet of shearwaters breeding in the Western Mediterranean Sea. So, thank you.
Thanks, Maria. Any question, please?
Hey, thank you very much for your talk. I was wondering, you didn't find any differences between population, but do you have any idea if the ratio between omega-6 and 3 have changed over time?
Over time?
Yeah.
Well, we only have one sample per bird, so we cannot.
Have you planned to maybe repeat such analysis during the next years?
Yeah, this is for my PhD, and I'm finishing, but maybe it would be interesting to see if there's a temporal trend, to see if these results are consistent, or because we analyze them in plasma, and as you know, plasma can vary, and it doesn't only reflect diet, but also fatty acids that are being mobilized for the different functions. So it's important to see if these differences are really coming from diet and then they are consistent, or if they are coming from another reason.
Thank you very much.
Any more? No? So, thank you, Maria. Now, now we're gonna have Aurore. We'll talk about overnight foraging, correct? Thank you.
Thank you very much. I think this is fine.
Okay.
And at 10 minutes I will show you the yellow.
Yeah.
And 12 I
the
red.
Perfect.
What?
Good day, everyone. Thank you for being here today, and thank you to the organizer for this lovely conference. My name is Rory. I'm a PhD student in my last year at Dehnhard University, Australia, as well as Ghent University in Belgium. So today I'm going to talk to you about overnight foraging trips in a diurnal seabird, the Australasian gannet. So we've had quite a few talks about gannets, so I'm going to take you to the other side of the planet to look at them. So seabirds in general are visual predators, which mean they're going to forage mainly during daytime, relying on visual cues. However, there are a few species of diurnal seabirds that have been reported doing overnight trips, so spending the night at sea, and potentially foraging at night. But for these species, spending the night at sea can be associated with some risks. For example, there can be a higher energetic cost to resting on the cold water at night compared to resting on land. There can also be a higher risk of predation by sharks, for example, or even fur seals. Also, there can be a higher risk of nest abandonment by the partner should the overnight trip last too long. And one of the species of diurnal seabirds for which overnight trips and foraging behavior at night has been recorded is the Australasian gannet. So this is a top marine predator in both Australia and New Zealand, where you can find about 53,000 breeding pairs. However, the prevalence, function, and drivers of overnight foraging in this bird has not been studied at all, which is not the case for all gannet species. It's been reported in, for example, northern gannets. But it can have implications for their adaptation to climate change. And that's especially important for the Australasian gannet because southeastern Australia is one of the fastest warming areas in the world. And it encompasses, as you can see on this map, most of the breeding range in Australia. So this warming has shown effects on prey distribution and availability in this area, and it can also have consequences on foraging strategies. And this is why, in this study, we're going to try and determine the prevalence and the characteristic of overnight trips, and we're also going to investigate which factors influence their occurrence. So the study was conducted in 2 breeding colonies in southeastern Australia, Northern Bass Strait. The first one is Point Danger. This is a coastal mainland colony that opens up in the open ocean with about 300 breeding pairs. The second breeding colony is Popseye, which is a smaller colony with about 180 breeding pairs located on an artificial island within a bay. So 2 types of GPS devices were deployed on breeding adults, iGetUs and path tracks, and they were both recording a GPS location every 2 minutes. They were deployed between the breeding season of 2011-2012 and the breeding season of 2023-24, during both incubation and chick rearing. Then a 3-state hidden Markov model was used to identify the 3 following behaviors: commuting, foraging, and resting. And we also added some accelerometer data that was available for only 3 breeding seasons to make the model more precise. A few definitions before we start on with the results. An overnight trip was defined as a trip starting on a civil day and ending on the following one, whereas a day trip started and ended within the same civil day. Nighttime was defined as being between astronomical dusk and astronomical dawn the next day, and that is because some gannets have been reported commuting when they had very little light between around civil twilight. So we really wanted complete night. So when we look at the prevalence of overnight trips, we have 12 breeding seasons of data that amounts to about 4,500 foraging trips. And 400 of those, around 500 of those were longer trips, where the birds spent more than 1 night on the sea. And those happened mostly during incubation and were more likely the result of the birds not being constrained to a limited foraging range by having to come back and feed their chick. So we removed those from the analysis so we could refocus on birds spending a day trip and birds spending 1 night at sea. And over the 4,000 remaining trips, 45% of them were overnight and 55% of them were day trips. When we look at the prevalence of overnight trips within individuals, day trips were present in 76% of individuals, while overnight trips were slightly more present, with 83% of individuals showing the behavior. And finally, when we look at departure and return times for day trip versus overnight trips, we see that day trips started in the morning and ended before nightfall, while overnight trips started in the afternoon and ended before noon the next day. Now we're going to look at activity budgets between, in the top row, the daytime activity budgets of day trips and overnight trips, and at the bottom row, the nighttime activity budgets of overnight trips. So as you can see, there is a higher proportion of time spent resting during daytime during overnight trips, to the detriment of both foraging and commuting. During nighttime, commuting was very occasional. Most of the time was spent resting, but there was quite a big proportion of time spent foraging, about 18%. Oh, sorry, spoilers. So now the question is, the bird that did overnight trip, did they do it accidentally because the day trip ran too long and they ended up too far from the colony and chose to spend the night at sea, or did they do it on purpose? So to have an idea of that, we looked at the mean commuting distance of foraging gannets, and we looked at where they were from the colony an hour before dusk. So the median commuting speed of gannets was 37.4 kilometers per hour, and we can see that about 50% of gannets doing overnight trips were well within the range of the colony. They could have totally come back. So now the question is, why and how are they foraging at night? So we're going to look at the effect of lunar phase on the proportion on the likelihood of a trip being overnight versus a day trip. And that is because the moon has been shown to have an effect, moon illumination has been shown to have an effect on the foraging at night of nocturnal species. So maybe it also have an effect on these guys. So 0 and 6, that's a new moon, so no moon at all, and pi is for the full moon. And as you can see, there is a higher likelihood of a trip being overnight between the first quarter and the full moon. So now we really want to see what exactly are these birds doing at night. So we looked at the accelerometric data that is available for 3 breeding seasons, and it separates foraging activity in 2 diving behaviors. The first one on top is plunge dive, so the bird is flying, it sees a fish, it dives for it. And duck dives, which is also called surface diving, so the bird is resting on the water and just diving from the surface. And as you can see, for plunge dives, there are 2 activity peaks, one at sunrise and one at sunset. But duck dives also show that peak at sunrise, but more importantly, they show diving through the night. So most of the foraging activity that is happening at night, all of it really, is duck diving. So now we are going to look at the effect of moon phase on this duck diving activity. And once again, you can see that duck diving activity is more likely to happen around full moon. And that is a really cool result. I'm really happy with that. Sorry. So another question is, why would they forage at night? They could very well do all of the foraging activity during the day, so why do they bother doing this duck diving activity at night? And one of the hypotheses is that they might forage on different prey. So to look at that, we have some isotope data that is available for, again, 3 breeding seasons. We only kept deployments that were at least 7 days long, and we only looked at plasma samples that were collected upon return after deployment. And we needed at least 80% of trips within deployment to be either day trips or overnight trips. So that way we could look at isotope ratio of nitrogen, so on the right side, sorry, which can be indicative of trophic level, and the isotope ratio of carbon, which can be more indicative of habitat. And in dark blue, you have mainly overnight trips deployments, and in light blue, you have mainly day trips deployments. And as you can see, there is a slightly lower proportion, there is a slightly lower nitrogen ratio in overnight trips, and a significantly lower carbon ratio in these overnight trips. So this is quite a small sample size, but models on a larger sample size also showed a very similar relationship, with overnight trips showing lower carbon and lower nitrogen, which could be indicative of them foraging on a lower trophic level. So to conclude, I feel like that was very fast. So to conclude, overnight trips are really prevalent within these birds. They make up about 50% of all foraging trips. And there's also a high level of nighttime foraging, which is really interesting in diurnal seabirds. Additionally, the behaviors appears to be influenced by moonlight, particularly duck diving. Considering the fact that plunge diving peaks at sunrise and sunset and the difference in isotope ratios, it's possible that its gannets are foraging on a specific prey type with a lower trophic level that could be being brought up by the diel migration of prey. And this is really important because the environmental changes in Bass Strait are really important, and they're likely to affect prey availability and distribution. And if these birds are not foraging on the same type of prey, depending on the type of trips they're doing, it might affect the choice in which strategies they're going to choose going forward. So thank you very much for your attention. I want to thank the Point Danger Committee, without which this research would not have happened. I also want to thank the 2 PhD students who collected and analyzed the isotope and accelerometric data, as well as the many PhD students and volunteers who sacrificed their knees to the gannets overloads. Many thanks to them. And if I have some time, just some shameless self-promotion. I'm in my last year of PhD, and I'm looking for a postdoc. Thank you.
questions, please.
Many.
We have 10 minutes to questions if you want.
Thanks so much. That was really, really interesting to hear about. I'm curious if there was any difference between the two colonies. You said one is much more
Yes.
facing the open sea and one is in a bay. If you can say something about that. Thanks.
Yes, that is a very cool question. Oh, sorry. Okay. That is a very cool question because these two colonies are very different in foraging behavior. So yeah, point danger, open ocean. So they show a bit less overnight foraging trips, which is really strange because of the bay. So the birds from Popseye, so within the bay, these guys, they have two very different strategies as well. So some of them are going to forage within the bay, and some of them tend to forage outside into Bass Strait. And the birds that go into Bass Strait have a very high proportion of overnight trips, but the ones that do forage within the bay are a much lower proportion of overnight trips, but they forage more during the night when they do overnight trips, which is very cool too.
Thanks, it was a really lovely talk. I'm over.
Thank you.
Is there any indication that your nighttime feeding might be in association with fishing vessels? And might that explain some of the nocturnal activity that they're assisted by the lights by the vessels as well as just the moonlight?
All right, I'm not sure I caught all of that, but fishing vessels and light potentially from them?
Yeah.
Okay, so there is a lot of traffic, maritime traffic within the bay and in this area. We don't have data for fisheries, so it's hard to know, but it could, like, association between gannets and fishing vessels have been shown, so it wouldn't be very surprising if the gannets in the bay at least enjoyed the light and like used it to forage.
Hello, here. Thank you very much. Really nice presentation. So do you have any data from other studies of the diet of these species, and any idea if they might be eating kind of more deep sea fish and mesopelagic? You mentioned that they could be taking advantage of this diurnal migration, because that pattern that you've shown is very typical of species preying on mesopelagic species. Any information from other studies about that?
So we do have some idea of what these birds are eating in general. So they like red bait, mackerel. They do eat some squid as well. So mostly schooling fish. From the isotope data we have, I can't tell what overnight trippers and day trippers are eating, if it's very different. But that's the kind of fish they're eating generally.
One more, please.
Thank you for the cool talk. I was wondering if you had sexed the birds and know if there's difference between males and females. So with the northern gannets, we found slightly more of the males being at the nest in the day, and they're potentially more aggressive. So they might be actually having quite a cost of being on the colony in the day, defending the nest.
Yeah. So that's really cool, because I'm currently writing the chapter and I'm referencing that study. So actually, females are showing more overnight trips compared to males. So with potentially males could be like more present at the nest and defending it more, while females could be going farther away and not caring as much. But yeah, females more overnight trips. Thank you.
Can you answer more?
Yeah, sure.
Sorry.
Okay, thank you very much for your talk. It's very nice. It's not a question, it's a suggestion probably. You mentioned that you have acceleration data, right? So it should be great, probably, if you can compare the energy budget from day trips and night trips. If you have the proper sampling frequency, you can take indicator of energy budget, and probably you have an explanation of that too.
Of a subpart of the paper. So one of the subparts is going to be looking at whether or not these birds are doing overnight trips because they're not foraging enough during the first day, and whether or not it's effectively like cost effective. And so potentially we might look at accelerometric data and compare between day trips and overnight trips and energetic budget. So yes, thank you.
Thank you, Aurore. So. So we're gonna start with the speed talks now with Lauren, correct? We talk about a little bit with genetic things now.
Hi everyone, I'm Lauren Evans. I'm in the first year of my PhD at Bangor University, and today I'm going to be talking to you about dietary metabarcoding and what Manx shearwaters have for dinner. So I just want to start with the importance of studying seabird diets. From a community ecology perspective, seabird diet studies help improve our understanding of marine food webs and whether ecosystems are governed by top-down or bottom-up processes. As we've heard, seabirds are also widely considered sentinels of ocean health, and changes in lower trophic levels are often reflected in their breeding successes and demographies. And in this way, they can also be useful from a fisheries perspective to help monitor fish stocks and populations. So there are numerous techniques currently used to study seabird diets, all of which have their uses and their applications, but all of which can be associated with different limitations. So stable isotope analysis is very useful for identifying broad-scale dietary change and trophic position, but it does only offer low taxonomic resolution data. Stomach content analysis involves either opportunistically collecting regurgitate samples or invasive stomach flushing, and so it's often not possible to perform repeat sampling, and often isn't applicable for species of conservation concern. So DNA metabarcoding of fecal samples overcomes a lot of these limitations, but it can be associated with low amplification success due to the presence of PCR inhibitory substances like urea in feces and degradation of DNA within the bird's digestive tract. And so in this pilot study, we trialed DNA metabarcoding of buccal swabs as a novel technique to overcome these limitations and determine the diet of Manx shearwaters. So buccal swabbing involves gently rotating a swab around the mouth of the bird, and it collects prey DNA before it passes through the bird's digestive tract. So in this pilot study, we deployed GPS loggers on 5 chick-rearing Manx shearwaters on Bardsey Island in North Wales, and then following a foraging trip, we swabbed both the adult bird and their chicks after feeding. For the DNA metabarcoding, we used 3 different primers targeting fish, cephalopod, and decapod DNA within our samples, and I'd be more than happy to chat to anyone later about the specifics of the lab work. This photo is clearly not a Manx shearwater. This is a northern fulmar and just shows the swabbing technique being used earlier this year when I collected a lot more samples. So in terms of results for the pilot study, I'm pleased to say that all 10 of our samples provided dietary DNA, which was identifiable down to the species level. So we use hidden Markov models to identify the foraging behaviour of the tagged birds using a combination of GPS, accelerometry, and dive data. And this gives us a rough idea of where the birds were actually catching their prey at sea. So this is an example of one of our longer trip birds. It was out from the colony for about 7 days, and it mainly foraged in the Irish Sea and around the Inner Hebrides. And I've paired this GPS track with the results of the diet data. So these charts show the results of the DNA metabarcoding, and they show the relative read abundance, which is the relative number of sequences of each prey type that we received in the adult and the chick swabs. So in this example, the majority of the adult and chick swabs were European sprat. The adult swab contained traces of whiting, and the chick swab contained low numbers of European pilchard sequences. This chart now shows the overall frequency of occurrence, which is presented as a percentage and is the number of adult and chick swabs that contained a given prey item. So you can see we detected European sprat in all 10 of our adult and chick samples, but we also picked up Atlantic herring, lesser sand eel, whiting, and European pilchard. So you can see there's a bit of discrepancy between the adult swabs and the chick swabs. And in this case, we think it may be because the chick swab represents prey items from more than one adult, as we can't be 100% sure that only the GPS bird fed the chick in this case. But this is something I'm controlling for in future data collection using geolocators to look at nest attendance rates. So in conclusion, I hope this pilot study has demonstrated that buccal swabbing and DNA metabarcoding is an effective technique with which to determine the diet of Manx shearwaters. The swabbing is minimally invasive, which allows for repeat sampling, and the metabarcoding offers high-resolution taxonomic data. And I think it has the potential to be widely applied to lots of other seabird studies. The next steps for this project and my PhD are looking at whether we can explain any spatial and temporal variation in diet using the oceanographic features that birds are targeting on their foraging trips. Thank you very much for listening. I'd like to thank my supervisory team, collaborators and funders.
And questions in the end, please. And now Lara Cerveira with Stalking Terns.
Hi, I'm Lara. I'm a PhD student, and I will talk about the spatial and trophic ecology of an estuarine bird. Lagoon and marine coastal habitats are very important for biodiversity and provide many ecosystem services. So, coastal and estuarine birds, such as little terns, choose beaches as breeding sites due to the proximity to the coast. But these areas are also increasingly used by humans, and human activities have negative effects on birds. For example, in this study, the authors also found that young European oyster catchers had reduced food intake and under human disturbances. But environmental factors as tides also appear to influence foraging behavior due to changes in prey availability, as we can see in this another study. So our main goal is access to tailored foraging patterns of little terns, habitat use, and trophic ecology. The study area was Ria Formosa in Algarve. That is a lagoon system, and we focused on two similar colony sites of little terns, Praia de Faro and Fuseta, both on sandy beaches, facing foraging coastal marine areas and adjacent to a estuarine lagoon system. For the first time, we used the mini GPS devices during the incubation period of 2 years in two different colonies. Nests in Faro and in Fuseta were marked and followed to measure hatching success. And we also collected regurgitates to identify otoliths and scales of prey consumed, and blood samples to measure health parameters and carbon and nitrogen isotope values. We also installed 7 cameras in each colony during the incubation period, monitoring 40 nests at each colony. And while at Faro, people, cats, hares, and dogs were responsible for the predation of 34 nests, at Fuseta, only 4 nests were predated. We found that Fuseta, a colony with comparably lower human disturbance, always had higher hatching success than Faro, in which the percentage of disturbances was almost triple. However, we found no differences in health parameters between colonies. Individuals from both colonies forage in areas surrounding their colonies. However, the maximum distance traveled from the colony was hiked by birds from Faro. Individuals from both colonies forage mostly during the day. Birds from Faro made longer foraging trips and forage more at lagoon and entrance channels, while individuals from Fuseta forage more over entrance channels, salt pans, and ocean. Birds from Faro preferred high tide, while birds from Fuseta preferred low and receding tides. And the diet was dominated by gobies and sun smells. However, we found more sun smells in Faro and more gobies in Fuseta. Also, we found no differences between colonies in the isotopic niche and the carbon and nitrogen isotopic values, but terns from Faro were more generalists. So overall, health parameters were low and similar between the two colonies, showing no visible signs of stress or inflammation. The differences in foraging habitat use between colonies might be due to different advantages in different tidal phases and different habitat characteristics. Individuals mainly forage during the day because probably the better visibility. Birds from Faro forage more in the lagoon channels, which explains why more sun smells were found in their samples. Individuals from Faro were more generalists and exhibited lower carbon isotopic values, which might be a reflection of a foraging over more pelagic areas. And maybe a solution for coastal seabird conservation could be implementing ecotourism practices to minimize negative effects on birds and their foraging habitats.
Thank you, Lara. Perfect on time. And now, sorry. Now, Astrid with the 5 costs. Dive or not to dive?
Hi everyone, so I'm Astrid Dedieu. I'm a PhD candidate at University College Cork. I know my abstract mentioned Manx Shearwater, but I didn't have time to include it all in 5 minutes, so I'm just going to focus on puffins, which are in fact the real best birds in the world. We try to identify wiggles in puffin dive data. What are wiggles? Wiggles are used to identify, they're used as a proxy for foraging success in large marine predators, large swimming marine predators, like elephant seals, seals in general, and penguins. And they're basically sharp changes in the trajectory. Imagine a seal swimming along in a fairly smooth line, and then it sees something and it tries to chase it, and it creates wiggles in the data. So puffins are pursuit diving birds. They do actively chase prey, and they spend a lot of time underwater. So we wanted to see if wiggles could be used on puffin data to make the same kind of inferences. Couldn't they be used here couldn't we identify wiggles first in puffin dive data, and what does it mean? So how did we do this? We tagged 34 puffins over 3 breeding seasons from Skellig Michael in County Kerry. You probably heard of the Skelligs from my colleague yesterday. And we tagged them with dual TDR plus GPS tags. We had an 80% to 100% tag recovery, which my supervisor was very proud about and told me to include in the presentation. We ended up, over the 3 years, with 16,218 dives, and that was for 24 individuals. We lost tags and some malfunctioned. We did have to cut out dives with less than 18 points for further analysis, and that's because we used a broken stick model that was built by Yves Legras, and that had elephant seals in mind, and it didn't really do well with shorter dives. So we ended up with 10,400 dives-ish. And so what the broken stick model does is basically breaks up your dives into sections. And then we built a custom function to identify wiggles within each of these functions, within each of these sections, sorry. So then I kind of defined a very arbitrary, simplistic bottom phase representing 80 to 100% of the maximum dive depth. And that was so that I could visualize where the wiggles happened along the dives, whether it be on the descent, the bottom, or the ascent phases. So we ended up with 8,121 wiggles across a little over 5,500 dives, which is about more than half of the dives we analyzed and a third of the total dives, because we did have to cut some out. These are just example dives, and the function identified these as wiggles, which looking at it visually is something that I would agree with. We characterize wiggles as being a change of the original broken stick trajectory and then a return back to that trajectory. So then we plotted where the wiggles occurred, and we saw that they appeared on the ascent majoritarily, followed by the bottom and very few on the descent phase. Here on the right, so that Y scale is flipped. I did only notice it this morning, I'm so sorry. So it's supposed to be 0 at the top and 100% down at the bottom. And that's it's depth, basically. So I did it as a percentage so that I could plot the wiggles to see where they occurred along the dive. And there's like a big clump at the bottom, and then they're scattered basically along the ascent with kind of a cluster at the very top, which represents the very end of the dive and the very top portion of basically near the surface. And we did see that a lot just when I was sifting through my dives. There were like this last little squiggle at the very top. Don't know what that means. So if we do assume that wiggles are a valid proxy for prey capture attempt in puffins, which is not what I'm saying yet, it supports the theory of foraging on ascent. Bottom face foraging or smooth dives with no wiggles in them could possibly be more targeted dives. I don't know. I'm just throwing ideas at you at the moment. We also have GPS location available for this. So I'm going to basically overlap bathymetry and seabed substrate just to see how deep the ocean floor is on those dive locations. Ideally, I would like to create an energetic profile of puffin foraging. My PhD is supposed to look at the impacts of offshore wind. I wasn't supposed to work on puffins, but the terns didn't cooperate, so I ended up with this data that was available. So I would like to basically incorporate this into a displacement risk model, hopefully. So yeah, lots to do with this data. This was just like the very first preliminary set of results. So, muito obrigada pela sua atenção. Thanks for listening. Big thanks to my amazing supervisors, which neither of whom are here today, and to all my amazing colleagues and ChatGPT. Thanks.
We take the sound off.
Yeah. Thank you.
Sound off.
Yeah,
Sound is off.
Okay.
Thanks.
Can I present you?
Yeah.
Yes.
Yeah. Now is Nina. We'll talk about fine scales foraging behavior, correct?
Yes, in European shags. And this is work together with my colleague Sven Håkon Lorentzen. So shags and kelp forests. We know that kelp forests are nursery grounds and hiding places for small fish, especially young gadolinids, and that also makes them important foraging places for seabirds. Threats to kelp forests include marine heat waves, outbreaks of sea urchins, which have devastated large areas of kelp forests in various parts of the world, as well as commercial harvesting to gain alginates. This is an aerial picture of a kelp forest that has been harvested. The European shag has a year-round coastal distribution. It's a species that is declining in large parts of Europe, but has a stable population in Norway. And I'm mentioning Norway because this work has been done in Norway. In this country, especially in the southern and central part, shags feed almost exclusively in kelp forests. We know this from previous work, and they feed mainly on young gadolinids. And my colleague Sina, who's also here today, has found out that there's a large spatial and temporal overlap in where the shags forage and where the commercial kelp trawlers go, which means, in other words, kelp trawling is happening in the middle of the breeding season and in the areas where the shags are going. And this is happening, for example, in Norway's largest shag colony, which is where we've been working at Sklinna. And that brings me to the research questions. Which parts of the kelp forests are shags actually using, and how much of the dive time is associated with different kelp habitats? So understanding better where the shags are in the kelp forests. So how did we do this? We did work on Sklinna, which is in central Norway. I did not add a map here, but you can, doesn't matter. In the middle of Norway. And we put loggers on 24 birds in the seasons 2020 and 2021 during chick rearing. And we put a whole set of 3 different loggers on these birds. A video logger from little Leonardo on the top of the back between the scapular blades. TDR, so temperature depth recorder, coupled together with the GPS with tape on the tail. So total logger mass was 60 grams, and of the 24 birds that we deployed, we managed to capture 22 back with the loggers attached. The videos lasted maximum 5 hours, and unfortunately, because we couldn't get the little Leonardo loggers to work quite as we wanted, we had to start them when we deployed the loggers on the birds, so I got a lot of nest footage as well to get through. So of those 22 loggers,
video loggers that we got back, 12 contained underwater footage amounting to 985 dives. We screened the images manually and did not use AI. I'm going to show a video a bit later and you will understand why I did not want to use AI. We excluded 28 open water dives where the birds never reached the ground. And we separated birds into the descent phase and the bottom phase, as well as the ascent phase. I'm not sure if the pointer is working, no. And the bottom phase we separated into whether they were in sugar kelp on the right, which is a very flat-lying kelp, which you can easily distinguish, and other habitats like sand, gravel, rock, or whether they were in these three-dimensional kelp forests that are formed by a species called Laminaria hyperborea, tangle. And for the tangle, again, we separated into whether the birds were above or below the kelp canopy. And this is the results. So 957 benthic dives. And this is only the distribution of habitats for the bottom time. I skipped the ascent and the descent phase. I did not have wiggles on the ascent phases in the shags, I can say. And you can see that the largest, the most time was used below the kelp canopy, top right corner, below the kelp canopy of the tangle forests. And you can see the stilts very nicely in the screenshot of the video. 21% of the bottom time pooled over all individuals was spent above the kelp canopy, and 27% of the bottom time was spent associated with sugar kelp. Gravel made up 17% of the bottom time, and sand and stone was hardly anything. So 53% of bottom time in total was associated with tangle, with that one single kelp forest species, and that's also the species that is harvested mostly. 80% of bottom time was associated with any type of kelp. And if I look at the individual dives, 83% of the dives were associated with kelp. So to conclude, and here is a video, and you can see how fast everything is happening. This is in real time. Our data highlight the importance of kelp and especially of tangle for shags in Norway. We also show for the first time that shags forage both above and below the kelp canopy, which is quite interesting. And the next steps is to publish these data. I have also coupled the data to TDR and to the locations, but there is no time to present this in 5 minutes, unfortunately. The next step is also to quantify habitat use and dive behavior in harvested versus pristine kelp forests. And with this, I would like to thank you for your attention. All the field assessments and
Thank you so much. And now, the last talk for this evening, Leticia, with fine costals Bermuda petrels. Yes,
thank you.
You're welcome. Good luck.
Thank you very much. So the foraging ecology and movements of gadfly petrels is poorly understood compared to larger seabird species. In this study, we try to get new insight at sea behavior of the Bermuda petrel, which is a gadfly petrel endemic to the western North Atlantic, and we try to answer 3 questions. So what is the diving ability of this species? How do petrels spend their time during their long foraging trips? And how does petrel activity change in relation to light and dark conditions? So to do that, we combine the use of 3 different logger types that we deployed on 25 different breeding individuals. And type A enabled us to collect 3 axial accelerometer data and pressures data. Type B was common GPS sensor, and type C had the characteristics of the previous 2 devices. So as you can see from the last 2 columns, we were not able to retrieve all the devices, so our data set unfortunately was not very big. So the first things we did was to map the foraging distribution of the Bermuda petrel during the incubation 2023, which overlapped with a very large area of the western North Atlantic. And then we focus on the diving behavior of these individuals in this area. So our results seem to suggest that petrels have a very limited diving capacity. From this histogram, we can see that the gray column represent the 99.99% of all dive observation, which had a depth that was less than 0.1 meters. So we just have one dive that gets the depth of 1.5 meters. The capture of the mesopelagic prey, which dominate the diet of this species and that migrate toward the surface of the water, probably must occur in the very upper layer of the water. So then we analyze accelerometer data, and we get a behavior classification in which we identify 3 behaviors non-flapping related to the flying activity, and they were non-flapping flight, flap, glide, flight, and the category that we called flying intensive that probably represent activity like takeoff, landing, or any aerial maneuvering. And then other 3 categories related to the activity of the birds on the water, when the bird was inactive, was active, or active water intensive, that probably also represent like bathing or preening or activity related to dipping the head into the water or catching a prey. We have to say also that it is known for these species that they are known to forage in flight, and so it is plausible to think that in the 3 flying behaviors could include foraging activity. So we analyzed the activity budget of these birds, and what we found is that petrels spend more than 3/4 of their time in flight, with non-flapping flight being the most common behavior in all conditions. So this is in line with the dynamic soaring flight strategy used by the species and in general by Pterodroma. And the rest of the time was spent on the water, either in active or inactive behaviors. And finally, we also try to see how the proportion of time spent in each behavior change in relation to light and dark condition. So the plot highlighted in this plot represent the predicted effect of sun angle on each behavior. The ones highlighted in red are those with significant relationship, and for example, we can see that from the first 2 plots on the top, that the proportion of the time spent in flying or flap, glide, or non-flapping flight has a strong negative relationship with solar angle, indicating that the birds are primarily aerial during the night. So I forgot to say that the gray area in the plots represents the dark condition, and the white area are the daylight. And if we have a look to the other 3 plots, the ones below that are related to the 3 behaviors related to water activity, so the 3 were positively related to associated with solar angle. So petrel seems to spend a significant time on the water during the day. These data are support and are in line with the expected nocturnality, marked nocturnality that Pterodroma species are expected to show. So thank you very much. We would like to collect more data, and if you have any question, please talk to me.
So we have time for 3 questions. Please, the speakers for this section can sit there, and we can start now.
Okay, thank you very much. I have an issue with an issue. No, it's just a comment for the wiggle talk. You mentioned that the wiggle was very good indicator of prey capture in penguins, mainly for fish eater penguins, which is sure. But probably you can also use accelerometers to corroborate that point in your birds. And it should be great if you can deploy two kind of accelerometers, one on the head and the other on the back, because this is really incredible, you know, process to understand the function of the wheels. This is just a suggestion and it's easy to do.
I'll just, we had these tags and it was very opportunistic. They were like, we have these tags, we're going to study puffins anyway, do you want to come and put these tags on? So that's what I did and I just worked with it, but yeah, absolutely. It'd be great to have more data.
Thank you.
Hello, thank you for all your talks. I've got a question for Lauren. We used metabarcoding for analyzing the data of ephesots, so a small wader. And we found quite a lot of funny DNA in it. So they were supposed to eat cats and Bengal tigers, if I remember correctly. And which, yeah, finding a cat in a poo sample is can be done, but a Bengal tiger in the Netherlands not. So, and I was wondering how much of funny DNA did you find in your samples?
Yeah, I had similar. I had like golden eagle, pig, dog, budgie, lots of random things, but none of them luckily made it past the filtering step. So we filter sequences. So our filter was 100, and it has to be more than that for it to be considered like a proper food item. So.
Yeah, and for those things like pigs and stuff, it's quite obvious that it's not part of the diet. But yes. Do you have some sort of mechanism to rule out things that might be in the diet, but are in fact not? Did you have made any progress on that?
Sorry. Yeah, we had a few species of crabs which potentially could have been eaten by the Manxies, but they were also at quite low levels, so we think it's more likely that the crabs were eaten by the fish which were eaten by the Manxies. So yeah, secondary predation is something you have to consider, but because they were at quite low levels, we think that's more likely, or at least they don't represent like a major food item for the Manxies.
Last question.
Okay, thanks. Actually, just as a follow-up question to that, Lauren, I wondered if you, and maybe you said it and I missed it, but did you also do the metabarcoding on fecal samples to see if there was a match between what you're seeing in the buccal samples and in the feces?
I didn't for this because we did think about it, but the idea was we wanted to look at like this meal that they're feeding their chicks, whereas if we took fecal samples of an adult, it would represent a different meal. So we wouldn't know if they were different because they're different meals or because they don't match up. So it's not something I did in this pilot study. Yeah.
So many thanks.